Director Oldenburg.
Board of Commissioners. April showers bring May floods. With May floods come mosquitoes. I thought this would be a good informational item to bring to your attention. Those of you who don't know, this is Tom Vallette. He's our invertebrate ecologist for the Forest Preserve District. He's been doing a diligent job with respect to the West Nile virus monitoring program. I thought it's important to point out that you're going to hear some talk in conversation in the public about mosquito abatements, and that is not what the Forest Preserve does. What we do is a program specifically cited at improving information for public health relative to West Nile virus itself. And so with that, Tom's got a brief report and can answer all your questions.
Thank you. Good evening. I'd like to thank you all for inviting us here to speak on this topic tonight. President Parati, Board of Commissioners, thank you. As Director Oldenburg said, I'm going to be giving you a brief update on our West Nile mosquito monitoring program. So prior to the start of this program, the district had been monitoring general mosquito populations in DuPage County for several years using contractors. However, in 2002, we had a very bad year. In fact, West Nile virus hit our state. We had over approximately 884 human cases and 66 deaths. Of that total, we had 52 human cases in DuPage County. 3 of them were deaths. So as a result of that, the Forest Preserve District started an in-house program in 2003 with particular focus on protecting public health. And the way we chose to accomplish this is to pick our target. We chose to target the Culex group of mosquitoes. And the reason why we chose that group of mosquitoes is that they are the most abundant group of mosquitoes in Illinois that are able to transmit the virus to humans and other incidental hosts. We are not targeting floodwater mosquitoes or other mosquitoes that are not able to transmit this virus.
How many mosquito kinds are there?
Many other mosquito kinds. I'll expand on that though, because that's a good question. Typically, when you think of a large rain event and you have 10 to 14 days later, you have a very large hatch of mosquitoes, those are not the mosquitoes that we're going after. We are going after container breeding mosquitoes, not floodwater mosquitoes. And Culex are those container breeding mosquitoes. So essentially, the first part of our program was to reduce breeding sites. We set up a series of monitoring locations throughout the entire county. You can see that here. That's just a very brief map. We also asked our division staff and our facility staff to take it upon themselves to reduce human-made breeding locations. And here's an example of what those are. Truck beds, boats that are tipped up right side up, leaky old gutters that haven't been cleaned in a while, used tires. Those are the types of human-made breeding habitats that Culex really like. And so our division staff and facility staff they have been doing that kind of source reduction ever since the inception of this program, and they still continue to do that to this day. Now, our second part of our program is mosquito monitoring. And so essentially the approach that we take is we monitor 2 different life stages of the Culex group of mosquitoes. We monitor larvae, which, as you all are probably already aware, the larvae are the aquatic form of mosquitoes. So typically what we're doing is when we monitor aquatic larvae, we're going out to areas where mosquito larvae are found, and they're typically found in areas where there are a lack of predators. A good predator on mosquito larvae might be a fish, for example. So we're not going to areas where we are finding fish. Fish are actually very good at controlling mosquito larvae. We're going only to the areas that there are no fish or other types of predators on mosquito larvae. So we've already reduced the places that we visit by a considerable amount. And with that, we are searching for Culex larvae. So what we're going out to do is we actually take samples of water in these fishless areas, and we're collecting that water, bringing back to our lab and identifying any larvae that might be present in the water samples. If we find that we have Culex larvae present, we'll go back out to that site and distribute a biologically derived larvicide. In addition to that, we also monitor mosquito adults, and that we use this trap right here. It's called a Gravitrap. And it's specifically designed to trap container breeding mosquitoes. We make it attractive to the container breeders. All the container breeders really like stagnant, dirty water. So we use grass clippings mixed with water, or sometimes horse manure mixed with water. They love that stuff, okay? And it attracts female mosquitoes that have already taken a blood meal. They're searching for a place where they can actually lay their eggs. When they come to the tub, they get swept up in through the battery-operated fan motor inside, and then they get collected in this net. We collect those nets, bring those back to our lab, and actually can test those mosquitoes to see if they are carrying West Nile virus.
How long does the water have to be in there to get that whole process to happen?
Not very long. Yeah, it gets pretty rancid smelling after just like a couple days.
So I mean, that's how long it takes them to lay their eggs and turn into a...
It depends really on the breeding cycle that's happening at when you place the trap. Adult mosquitoes that are in the area that have already taken a blood meal will probably be almost immediately attracted to that trap. Was that your question?
I'm thinking about people's backyards, that if you have a planter that it rained 5 days ago and you dump it out, is that going to help
Yeah, that's
you?
a typically good turnaround time for control. However, if you're attempting to reduce sources like that, if it's hotter out, the mosquito development times decrease. So take, for example, a Culex might go from egg to adult under normal conditions, say 70 to 80 degree temperatures, probably from 10 to 14 days. But under 90 degree temperatures, that gets reduced from 5 to 7 days. So you're going to want to turn that probably more frequently than every 5 days.
But if that dries out, or
Yeah.
you dump it out, then it's...
you're good.
Then, okay.
Yep. But like, say, take for example, an unmaintained swimming pool. You can't very easily dump something like that out. That's why we use larvicides to control those areas that you can't easily dump.
And is that gravid mosquito trap completely different from the sticky cards that we see around other parts of communities, capturing insects?
Yes. Yes, it is very different. I'm actually not familiar with that. That may be more of an emerald ash borer collection mechanism. Typically what's done for collecting floodwater mosquitoes, the other types of mosquitoes that I was talking about earlier, those are usually collected with light traps or baited with dry ice because they're attracted to CO2. And so that's just a measure of getting general mosquito populations. So this is not anything near what that is. Does that answer your question?
Yeah.
Yeah, the sticky traps, I'm not actually familiar with that type of trap. So if we do test positive for West Nile virus, we can use that information in one of two ways. We can report that to local health departments. We do report our positive test results to DuPage Health Department. But we also use it for our own program too. So we can focus our larviciding efforts around a geographic location near a positive test. So here are some results from last year's program. We managed 165 sites, and of these, 135 were larval sampling sites and 30 were adult monitoring sites. We administered 252 tests for West Nile virus. 22 of those were positive. We had an 8.7% positive rate. Just to clue you in, that's probably the highest percent positive rate that we've had since the inception of this program. Typically, our percent positive is around 4 to 5%. So it just tells you something about how much virus was in the environment last year. Last year was pretty rough. And another indication of that were statewide. Human case numbers, 290 human cases, 12 deaths. That's the most number of human cases that we've had in the state since the 2002 outbreak. Of those totals, we had 56 human cases and 5 deaths in DuPage County. So here are some conclusions. We definitely know the virus was increased last year in 2012. We had hot temperatures, dry conditions, above average number of 90-degree days. In general, it was a, quote, bad year for West Nile virus. Now, we know weather patterns continue to be the determining factor in the appearance of the virus. In fact, that major drought that we experienced last year encompassed the entirety of the state of Illinois, and it did contribute to this effect. Culex mosquitoes breed primarily in containers, as I said before, and in stagnant pools. So drought actually tends to increase their presence and thus increase the prevalence of the virus. Now, that may be kind of counterintuitive, but as I mentioned before, the floodwater mosquitoes are the ones that respond more to the rainfall events. But Culex mosquitoes tend to react to human-made environments better than more other mosquitoes. In fact, Culex, the common name for the Culex mosquito is the northern house mosquito. That tells you something about where it likes to breed. Okay. And so in a drought year, you typically have changes in human behavior. People don't experience many floodwater mosquitoes. So what do? I'm not getting bitten. Okay, I guess I'm not going to wear DEET repellent. Okay. That increases the prevalence of you getting exposed to the disease. What else do people do when there's a drought? They water their lawns more often. Okay. That water, what does it do? It runs off into a stormwater catch basin. Perfect opportunity for Culex to breed. So as you can see, contrary to logic, Culex actually do better in drought conditions than in any other conditions. And I'll show you a little bit more about the conditions that we experienced last year. Sorry for the complexity of this graph. Really what I want you to look at here in this graph are the drastic fluctuations in temperature. The top line is in orange is the high temperature. The green line is low temperature. And the blue line is rainfall throughout from the beginning of our sampling season all the way to the end of the sampling season. And you can see these are these peaks up here are 90-degree days. And you can see there are parts of the season where there just was no rainfall, right here. And then in those particular spots of the season, you see Culex numbers, those are the red bars here, increase markedly, okay? So we had those conditions in Illinois, and on average, let me talk a little bit more about the 90-degree days. On average, we have 19 days in Illinois that are at or above 90 degrees Fahrenheit. Last year, we had 45. Almost a month and a half of at or above 90 degrees Fahrenheit. Okay, so that really drives the viral cycle when you have such huge hot temperatures. You get the urban heat island effect at night. Temperatures are hotter than they potentially would be in natural areas. So you have the compounding effect. So here's a graph that I like to talk about when people ask whether we should continue this program. And I've discussed this topic with my colleagues, other people who love sampling mosquitoes. And the response that we typically get is West Nile virus continues to return year after year. And this graph really shows that. What you're looking at here is percent positive tests from 2 sampling agencies in DuPage County. The blue is DuPage County Health Department, and the red is us, the Forest Preserve District. So these are percent positives. And what it shows is that at the beginning of the program, we had, considerably detectable levels of virus. But as you move on towards, 2007, 2008, and 2009, the Forest Preserve District, there were just not detectable levels of virus in the environment. And I remember going back a few years, we were wondering, oh, well, is West Nile virus going away? Got a lot of questions about that. And then came 2010. Right back up where we started at 2006 at similar levels. And then 2012 was really our worst year since the outbreak. So really, essentially, the takeaway here is West Nile virus has not gone away. It's not expected to go away anytime soon. In past years of the program, we've often looked at ways we can maximize efficiencies of the program. And 2013 is no exception. So what we did is looked at our program and found ways that we could maximize those. And one is an example where last year, we all know last year was a bad year for West Nile virus. Well, if we had a testing site where we were collecting adult mosquitoes and that did not test positive, we deemed that site as not being sensitive enough to detect West Nile virus in the environment. And so we eliminated that site. If it wasn't going to test positive in a hot year, chances are it's not going to test positive right now, you know. So we eliminated those sites from our program. In addition to that, sites that were in close proximity to other trapping agencies in the county, we eliminated those sites as well. So as a result, we came up with some management recommendations for this year. Sorry, it's a little bit tough to read, but we're going to be sampling and treating 135 dip sites for Culex mosquito larvae. We'll also be monitoring and treating stormwater catch basins on our property. We have approximately 300 stormwater catch basins within on our property, mostly in parking lots, to reduce mosquito development. We'll also reduce adult mosquito sampling sites to 6 locations. The sites that remain in the program are sensitive to West Nile virus and do not overlap with other agencies. We'll continue our historical database and continue working with Illinois Department of Public Health, sharing data as well as sharing data with DuPage County Health Department. We'll also analyze trends from past reports and compare with our current data, and continue searching the scientific literature and interviewing some of my colleagues and other experts to determine future directions of the monitoring program. And with that, I'll be happy to take any questions that you may have.
What other counties do monitoring like this?
What other counties?
What other county forest preserves do monitoring like
I
this?
think we're the only ones, actually. There are other county forest preserves that have contracts with companies to do similar types of monitoring programs. However, I think we're the only ones that have an in-house program.
And do we use any volunteers for the collection of the data?
No.
Do you see that that might be possible someday?
The reason why we haven't used volunteers in the past is because typically what needs to happen when we're collecting data, we're also using larvicides, and you need a state license to apply those larvicides. So in terms of efficiency, we've just used paid staff to accomplish both tasks.
Is it an herbicide license or is it something different?
It's basically the same thing as what you know as an herbicide license. You have the general standards.
Thank you.
Sure.
As a layperson, you say that the flood mosquitoes don't carry the disease, but the
Well, they
Culex
carry
does?
the disease, but they cannot transmit the disease. It's different. They are actually physiologically different than Culex, the Culex group of the mosquitoes. Their salivary glands do not operate in the same way. So that when they take a blood meal, they don't actually inject you with some of their saliva, whereas Culex does, and that's how they're able to transmit the
If
virus.
we're at a picnic in mosquito lands, we would in no way know what we had on our arm.
Exactly, that's why repellent is so important, to wear repellent. Yeah.
And our sites have gone down from 165 to 6, there was that much overlap?
No. 165 was the total number of larval monitoring sites and the total number of adult monitoring sites. So last year we sampled 30 adult monitoring sites. That's getting reduced down to 6. So our total monitoring, I think what was it, 135 or something like, that's our total number of monitoring locations, including the adult monitoring sites. Actually, that's not including. 135 larval monitoring, 6 adult monitoring sites, so a total of 141.
We approved the contract with Clark Environmental just moments ago.
Yes.
The $53,000, is that, do they do anything with regard to that, or are they more contracted to do?
That is
Abatement
strictly
needs.
aerial herbicide application.
Okay.
This whole issue started years and years ago because, as Tom indicated, Clark Mosquito used to do monitoring countywide in the forest preserve. And then we found, unfortunately, that they were treating large areas of the forest preserve with various insecticides, and we put an end to that. We looked at then this West Nile virus program contractually, and they could have done that program, but it came out to almost twice what our staff could do. And thus was born our recommendation to start a staff program. And I have constantly challenged Tom because his expertise is needed in a lot of different areas, and we'd like to see, spends a considerable time with West Nile gypsy moth, and certainly would like to see his talent spent on desirable species as well. Constantly asking.
I like mosquitoes, I don't know what you're talking about.
You said that there were about 300 catch basins on our property?
Approximately, yes.
How many of those do you typically treat? Now, are you only treating certain areas that are susceptible, or
We
how
are
you?
treating all of those catch basins.
Okay. So if constituents
Right.
ask us if we're treating them, so that's, we
Precisely.
can tell them.
Any other questions?
Do we treat for emerald ash borer?
We do not. We do not treat for emerald ash borer, that's correct.
Okay, well, thank you very much for the very
Thank you.
informative presentation.